I'm Kit and I'm a somatic cell. I'm made out of candy. Today I will be performing mitosis in preparation for Halloween to make more candy. Mitosis occurs in somatic cells and allows sexually reproducing organisms to develop from single cells and offers repair and renewal of old cells. Cell division passes genetic information to future generations. The cell cycle begins with interphase. Here, the genetic material of the cell is present in the form of chromatin. In the first subphase, G1, the cell grows. Next, in the S phase, the chromosomes duplicate and the cell continues to grow. Lastly, in G2, the cell finishes preparing for cell division. Prophase is the first step of mitosis. Chromatin fibers condense into discrete chromosomes. The nucleoli disappear and the nuclear envelope disintegrates. The replicated chromosomes appear as two identical sister chromatids. The mitotic spindle begins to appear and centrosomes move apart. In metaphase, centrosomes reach opposite poles and the mitotic spindle forms. The microtubules of the spindle attach to the centromeres of the chromosomes and transport them to the metaphase plate. Anaphase involves the separation of sister chromatids. Each chromatid is now considered an individual chromosome. The spindle microtubules begin to move the chromosomes to opposite poles of the cell. In telophase, the last step of mitosis, two daughter nuclei begin to form at opposite poles. Nuclear envelopes and nucleoli form, and the chromosomes decondense. Cytokinesis usually accompanies telophase and involves the division of cytoplasm. In animal cells, a cleavage furrow is formed, eventually yielding two identical cells. In plant cells, a cell plate divides the two daughter cells. Each resulting daughter cell has two N chromosomes. As you can see, mitosis is very effective. However, if one is looking for variation, one should use meiosis.
Hi, my name is Kat, and I am a germ cell. I, too, am made out of candy. I need to perform meiosis in order to make more candy for Halloween, and I plan to make many kinds. Meiosis is very important because it results in genetic variation, which is key for the survival of a species over time, especially considering changing environments and natural selection. A very important factor of meiosis is that it reduces the number of chromosomes so that the resulting cells have N chromosomes. This is essential when gametes fuse to create a zygote. Thus, this zygote will have two N chromosomes, one half from each parent. Meiosis consists of interphase and two cell divisions, meiosis 1 and 2. In meiosis 1, homologous chromosomes separate, resulting in two haploid cells with replicated chromosomes, while in meiosis 2, sister chromatids separate, resulting in four haploid cells with unreplicated chromosomes. Let me show you how meiosis works. During interphase, homologous chromosomes replicate. Prophase 1, chromosomes condense and homologs pair. Crossing over occurs between non-sister chromatids, in which segments of DNA are transferred, and this occurs while homologs are in synapsis. The nuclear envelope breaks down, centrosomes move, and spindles form. Homologous pairs migrate towards the metaphase plate. In metaphase 1, centromeres attach to microtubules. Homologous pairs are lined up along the metaphase plate, with the chromosomes of each pair facing opposite directions. In anaphase 1, homologous chromosomes separate, while sister chromatids remain attached and move together to the same pole. In telophase 1, each side of the cell has non-identical, replicated N chromosomes. Cytokinesis occurs and two non-identical haploid daughter cells are formed. Now begins meiosis 2, where the succeeding steps occur in both daughter cells simultaneously. In prophase 2, the meiotic spindle forms and chromosomes move towards the metaphase plate. These chromosomes consist of two chromatids. In metaphase 2, microtubules from opposite poles attach to the centromeres of the sister chromatids, and the chromosomes line up on the metaphase plate. In anaphase 2, sister chromatids separate. The now individual chromosomes move to the opposite poles of the cell. In telophase 2, nuclei form and chromosomes decondense. Cytokinesis follows and four non-identical daughter cells, each with unreplicated N chromosomes, are produced.
Before we go, I'm going to compare and contrast mitosis and meiosis. DNA replication occurs in interphase of both processes. Mitosis has one division and meiosis has two. Crossing over and synapsis occur in meiosis, but not in mitosis. Finally, mitosis results in two diploid daughter cells that are identical to the parent. However, meiosis produces four haploid non-identical daughter cells.
We're gonna take a break now, but we'll give you a piece of that Kit Kat bar.